Well, good afternoon. We are going to start the next session on stem cells for disease modeling. So we have today 3 different speakers, and we are going to illustrate advances in the technology of disease modeling, including toxicology, obviously, using 2 specific disease areas, cardiovascular and neuronal disease. But of course, those tools can be used for many other types of indication. So the first slide here, I just wanted to illustrate here the evolution of the drug design. How did pharmaceutical companies have developed the drugs that we use today? And it's important to keep in mind to really understand the power of the new technology here. So essentially, let's not also forget that until the 1800s, the pharmaceutical industry was essentially a little more than plant extract. So we have done, over the past 100 years, and particularly in the past 20 years, an enormous amount of progress. So we started from very, very early, where we wanted to first match a compound and some particular effect. That was a paradigm in the early 1980s, I would say. And then we started to develop a lot more sophisticated tools, matching essentially activity and drugs and particular targets, and even more to do target design compounds. But all of these have limitations, and this limitation is illustrated in the next slide, which is here. And that really represents the biggest problem for the pharmaceutical industry, which is the attrition rate. And by that, what is meant is the number of compounds that fail as we progress in R&D and clinical translation. And this is just a very old slide, but that really illustrates the problems of the 1990s that we had. And this issue of attrition rate has resulted in skyrocketing cost of drug development. So the new tools really provide us a chance for the first time really to have essentially a human and a chip, human in vitro, where we can assess the activity, the toxicological properties of new compounds much more accurately than ever before. Because a mouse cell is great, a line of mice is great for disease modeling, but at the end of the day, anything that is closer to humans is better. So I'm going to leave Dr. Timothy Nelson speak about applications in cardiovascular disease, and I'll let Tim present in a few words what it is you have done so far. What is your vision for disease modeling using stem cells in general?
Great, thanks for the time and the invitation to speak today. It's a great honor to be here and share with you some of our experiences with induced pluripotent stem cells and disease modeling in a congenital heart disease known as hypoplastic left heart. I'd first like to thank the Todd and Karen Wanick Family Foundation that has supported this work and has really allowed us to take this to a new level and start playing with this technology to try to make it a pipeline for us to do drug discovery and disease discovery mechanisms of hypoplastic left heart. My disclosure is Regen Theragnostics, which is a fee-for-service provider of induced pluripotent stem cells that Mayo Clinic and myself have financial rights to. So hypoplastic left heart is a rare but severe form of congenital heart defect that the left side of the heart is absent or devoid when they're born. These children require 3 open heart surgeries at 3 days, 3 months, and 3 years of life. This surgery is designed to cut the left ventricle out of the circuit and make the right ventricle do the entire workload of the body circulation and the pulmonary circulation. This 5-horsepower engine inevitably fails and leads to the need for heart transplantation for those that live long enough to get that as an option. Because of this severity of this disease and due to the surgical innovations in the last couple of decades, the survival rate has progressed from 0 in 1982, progressing to nearly 50% of these kids living to their 5th birthday. 50% of these kids living to their 5th birthday today is still a catastrophe And so the question of where does regenerative medicine and stem cell therapy come into play to try to rebuild these congenital defective hearts as we go forward. We have ongoing clinical trials using stem cells in this space that's currently being done at Mayo Clinic. But for the focus of today, I'd like to talk about how we use induced pluripotent stem cells to try to model this disease in a dish to better understand the mechanism of disease to ultimately find new curative and diagnostic solutions. So just as an overview for this session, induced pluripotent stem cells, you all know what they are. They essentially are bioengineered stem cells that can come from a somatic tissue such as a skin biopsy. And by reprogramming them with viruses or proteins or small molecules, we can get these fibroblasts that grow in your skin to convert into induced pluripotent stem cells that have the capacity of differentiating in all 220 lineages of your body. So this remarkable ability to have pluripotent stem cells that are bioengineered from yourself, makes them autologous, is a true game changer. This game changer only became available in 2007 when this was done for the first time in human cells, and now that has transformed the way we do biomedical research in the discovery sciences arena and will ultimately transform the way we do diagnostics and ultimately therapeutics. It allows us to have these cells that grow indefinitely in cell culture, to do genetic engineering on them. It allows them to differentiate into any of the lineages we might be of interest, and specifically in our case, it's the cardiovascular lineage. And it allows us to understand what's going on in your body when you were a few weeks old as an embryonic stem cell. So this profound game-changing technology is real, is available to us, and now the challenge that you and I are facing every day is how do we apply that? How do we leverage this for discovery sciences and disease modeling? So some of you have seen these types of pictures before. Hopefully the movie plays here. Could you click on the video itself on the left-hand side? Thank you. Or the play button. So this is cardiac tissue that has been engineered in 13 weeks after having a biopsy. Starting from a piece of skin, you can create these hundreds and thousands and millions of cells that work together in a system of cardiac tissue. It's not only a two-dimensional structure, but you can, and hopefully this one will play as well, if you can click on the movie. Not available. That always happens once in a while. All right, well, envision this heart-like structure that is actually in front of you here. It's actually contracting. If you don't believe me, we can show you after it on my own computer. But this is about 3 million cells that are working together and they're literally contracting like this. It's a three-dimensional structure that's pumping fluid out of the connecting conduits on the right and left and on the top of this construct. And it's working in a system of cells with the heart muscle contracting together. But you're looking under a microscope at about the tip of a ballpoint pen on this. So it's clearly a small structure, and people will ask us, well, how did you do that? The reality is we did nothing. The reality is that these are pluripotent cells that when you allow them to differentiate in culture, they can form cardiac tissue like you did as the first organ when you were an embryo. And we don't fully understand this process today, but we are getting much, much better at being able to make this consistently cardiac tissue from virtually any patient that we biopsy. Now this started out being a one cardiomyocyte that would contract. We were excited about 10 years ago. And now we have the requirements of making 70, 80, 90% of the cells in the culture beating, contracting cardiomyocytes. But what's really remarkable for me in this technology is that this isn't just a tissue that we've bioengineered. But if you put adrenaline on these cells, you can make it contract twice as fast. If you put a beta blocker on these cells, you can slow it down. So these cells that are in culture that came from your body are actually pharmacologically responsive to the same drugs that we would be giving you. This allows us to test drugs on your heart without ever giving you the drug. And this is a game changer and really allows us to envision how cardiac toxicity drugs could be tested today for things such as long QT syndrome. So today what we have accomplished is we've biopsied nearly 200 patients now. As stated here as 120, we've done about 200 patient biopsies. We've converted that into about 700 different iPS cell lines, and we now have this on probands with hypoplastic left heart and moms and dads that do not have disease to be able to compare. So by having that trio of samples from hypoplastic left heart individuals and moms and dads that don't have it, it allows us to compare the cardiac tissue head to head with whatever biological or pharmacological assay you might have available to you in the lab. This large repository of iPS cells also has whole genome sequence on every one of these patients. So it also allows you to search for genetic causes of this disease, find a potential pathway of interest, and then zoom in on the iPS cells or the cardiac tissue to determine if those pathways are normal or abnormal in these cells. This allows us to have a genotype-phenotype relationship with patient-derived cardiac tissue in the culture like we've never had before. But it does beg one question: what is normal and what is abnormal? So in the context of congenital heart disease that I'm talking about today, we're going beyond the ability to diagnose the disease, which is quite easily to do with an ultrasound or an echo. We're even going beyond the disease modeling phases where you change the focus to studying the tissue, such as maybe a hypertrophic cardiomyopathy, maybe a long QT syndrome, where you could study the actual defective cardiac tissue. And we're going beyond to try to use this technology to focus on the process of developing normal heart tissue. The process of cardiogenesis that goes from stem cells to cardiac tissue that goes abnormal in utero for these congenital heart patients. Can we understand the defective pathways of that cardiogenic process in culture and try to understand on an individual basis of the patients what we could potentially do to intervene and correct that even in utero if we could understand it well enough? But how do you really know what normal is becomes a big question. It first starts with key attributes of these cells. You have to have high quality iPS cells. What is a high quality iPS cell? It's debated, and we have to really concentrate on this as a field and as a community to define what is high-quality cells. We have to have an embryo-defined normal process of what do we expect them to do if they were normal. And then ultimately, we need the genotype-phenotype relationships to be able to really understand if this is the pathway that's defective in the cardiogenic process. If we reverse one gene, one pathway, do we reverse the pathology in the cell culture? Is our definitive, necessary, and sufficient type of test to prove that we have the key for that individual patient? All of that was only possible in creating knockout mice, knockout animals in the past, and cell culture that we may not recapitulate these disease entities. We now can cut all of that out of the middle, and we can go directly to the patient-derived sample and try to understand what is normal and what's abnormal. And finally, I'll just show 2 slides that what we define as normal is we have done extensive work in dissecting out mouse cardiogenesis for decades, well beyond one lab and one institution. And by doing this dissections of normal heart development in mouse embryos, you can profile this with genotype, proteomic, or metabolomic type of profiling. And in this case, we used RNA profiling to create these roadmaps of every single gene that's expressed in normal heart development at different stages. So we can watch the transition from genes of pluripotency transitioning into cardiogenic at various stages of development. And plotting those, all 50,000 genes that are differentially expressed in bioinformatic tools to see the transition of these genes creates the normal roadmap so that we can then compare the patient-derived samples, whether they're normal or abnormal. And ultimately, the goal of this type of work, using this new type of platform, would be able to pinpoint the initial point of divergence. The initial point of embryogenesis that goes from normal to abnormal might be at few weeks of life, few months of life. And if we can identify that initial point of divergence of normal and abnormal cardiogenesis, that provides a very powerful target to go after therapeutic strategies to try to cure and prevent this disease going forward. So that's an overview and a summary, but this is one person that has the good fortune here today, myself, speaking on behalf of a large team of about 60 people that work in this program. And I'm grateful to all of the scientists and expertise that allows us to bring this multidisciplinary team together to apply this technology and hopefully find new curative solutions for this devastating condition of hypoplastic left heart. And I'd be happy to take questions later after the other 2 colleagues speak.
Well, thank you very much, Tim. So I think there is a first learning here. When we look back at the first slides that we had in the 1990s, the paradigm has totally changed. It's a brand new world here for the pharmaceutical research, because this type of understanding, to decipher the pathobiology of disease, is a brilliant, brilliant advance. And we can hope that with conventional therapeutics, this type of model will really help bring more efficacious and safer drugs. Now, we looked at cardiovascular, which is a very important system. Let's look also now at something more brainy. Let's look at neurological applications. And our next speaker is Fernando Pitossi, actually from the Leloir Institute. Thank you. You can pronounce it better than me. Thank you.
Thanks, Alain. It's a great pleasure to be here. Thanks for the invitation. So I'm a scientist. I work on a research institute that is called the Leloir Institute, and I'm also head of a platform to provide services for reprogramming cells and derived into neurons. That is called Plasema, and is publicly funded. So just to give you a bit on the environment we work in, we are very fortunate to have Minister of Science, and that is a former stem cell biologist. So he really knows what we are talking about when we are talking about stem cells. And he has promoted the creation of a consortium called CISEMA, also this humanoid place platform, and we have a very close relationship with Brazil through many cooperation treaties. So this platform is publicly funded. It's a non-for-profit association. I just wanted to make that clear because some of the results I'm going to show you are coming from the quality controls that have been made from this platform. So as Tim has just told you, there is a clear pathway now to try to benefit from that drug discovery based on human reprogramming, and that is basically by taking cells from patients, either fibroblasts, blood cells, or other type of cells, reprogram them, and then differentiate them into the specific cell type. And what we have been found, and many other groups in the world have found that it was that in many cases, especially if the disease is genetically based, the resulting cell from this procedure has features from the condition that one is studying. So it's a very nice way of making in vitro modeling of diseases from a patient that is still in the course of the disease. We usually use lentiviral vectors coming from the University of Boston, from Dr. Gustav Mostoslavsky, to reprogram. And I also wanted to point out that apart from using this technology for drug screening and to discover new drugs, one can also think of getting cells from normal population, not from sick patients, and try to do toxicity screenings from hepatocytes, neurons, or cardiomyocytes coming from that healthy population. And that's one of the main initiatives that the government of Argentina is trying to promote. But we are here to talk about disease modeling, and I'm going to show you some data from our lab that is specialized on Parkinson's disease, but I'm going to show you something about the epilepsy data we have produced. So this disease is the third most common neurological disorder that affects patients from all ages, and it's estimated that 1% of the population will develop this disease at some point in their lives. It is caused by an uncontrolled and hypersynchronized electrical activity. And the problem is that the available therapies act on the symptoms and not the causes, so it's really we are really need to have models of disease where we can try new drugs on and try to understand the disease better. The type of epilepsy we studied is called benign focal childhood epilepsy and represents the most prevalent syndrome in the pediatric population. And one feature of this disease is that as the children get older, the symptoms reverse. So it's affecting the childhood of these patients, but most of the times in the adulthood, the symptoms are reverted. And this is a very important feature. So we have studied this family where you see that out of the 7 children of the family, 5 of them are affected with different types of epilepsy, mainly this benign focal childhood. And we have studied or taken samples from 2 daughters that are affected, 1 daughter that is a healthy control, and also a sample from the mother. This is the workflow we use for reprogramming and differentiating the cells, and in the end try to get a model of, in this case, epilepsy. So in real terms, we take, in this case, biopsy under local anesthesia from a patient or a control, derive a fibroblast, then use this lentiviral vector to produce iPS cells. And one important feature for us is to define and be sure that when we say iPS cell, we are really dealing with the fully reprogrammed cell. And for that, we use 9 quality control assays to check for pluripotency, what we call stemness, the silencing of the transgene that was inserted, normal karyotype, of course, sterility, viability, and integration of the reprogramming information. And this is done by this human iPS platform. So once we have the iPS and we are convinced that these are real iPS fully reprogrammed cells, then we differentiate them to what people call mini brains or organoids from neural origin. And this is done by a protocol that takes at least 8 weeks. And during those days, you can see structures forming that if you characterize them, for example, by immunohistochemistry, you will see, as in the lower right panel, some reminiscence of ventricles and some cortical organization in these structures. And all these cells have markers, express markers of neural phenotypes. These neurons are also active in terms of electrophysiology, so they have sodium and calcium functional channels as detected by electrophysiology. And when we go to the characterization of the neurons coming from epileptic patients, we see that at 10 weeks after differentiation, the neurons coming from patients have still immature markers, so that they are not fully mature neurons, that they have also an increased cell death from apoptosis at an early time point, and that in the end, if you look at the right panel in the bottom, and that's a marker for glutamatergic neurons, specific type of neuron, and they have a reduced percentage of those type of neurons. In addition, we see that neurons coming from the skin of epileptic patients have a reduced axonal length, and they don't make synaptic contacts compared with the controls. So as a summary, I think this is evidence that one can take cells from an epileptic patient, reprogram it, differentiate it to a neuronal phenotype, that these neurons are alive, that we have a population of 60% of glutamatergic, 40% of GABAergic neurons, and that we can make a deep analysis on these cells and try to establish this in vitro model for more and deeper drug discovery. These are the people in the lab who did the work on the left. We have a lot of collaborations in the country, and also as in the advisory board of this platform, we are very proud to have Mahendra Rao, that maybe you all know, and Xianming Zheng, who is advising us on the neuroepithelium differentiation. Thank you very much for your attention.
Okay, thank you very much, Fernando. So let's ponder a few minutes here. So what have we seen today so far? We have seen two presentations in two totally distinct disease areas. And what we have, again, is a way to model disease. And I would like to restate, to emphasize how wonderful that capability can be, because epilepsy has really been one of the diseases that has been struggling, vexing essentially all the scientists, medical professions, I would say, for centuries, right? And we reach a point where with a few experiments in a laboratory, we can start understanding the fundamental basis of this disease. And that knowledge is precious, because with that knowledge, we can start suddenly hoping to find a cure. So this is important. Now we go back to cardiovascular indications to really illustrate how versatile the technology of embryonic and IPS models can become. So I would like to welcome Gary Smith from the University of Michigan. And Gary, if you can again provide what it is you want to achieve, what is your vision, and of course, give us a brief introduction of your work. And we'll have a few more minutes at the end of the presentation for questions.
Great, thank you very much for the introduction, and thank the organizers for allowing me to speak with you today, and I'd also like to thank all of you for participating in this session. My discussion today is going to be a bit different than the other discussants. Mine's going to be focused solely on human embryonic stem cells and using human embryonic stem cells to model diseases. I think one of the very first things to recognize that a real line of demarcation between human ES cells and IPS cells is that human ES cells are a great model for single gene disorders. If you know a certain gene causes a disease, understanding the progression of the disease with a human embryonic stem cell is possible. For IPS cells, you can address diseases that are not single gene disorders. So if I think about bipolar disease, human ES cells would not work for studying bipolar disease, but an IPS cell will work for studying bipolar disease. So that's kind of a beginning of the talk. So quickly, just a reference to the people in the laboratory who really allow me to come and talk to you. These are great people that I work with. There's about 60 years of human embryology, IVF experience within these people, and it's one of the things that contributes to our success of making human embryonic stem cells for disease models. So how do we get human embryonic stem cells? Well, there's two different ways. First, you can have a family that has infertility. They go to see an IVF doctor, they produce embryos, they want to build their family, they transfer 2 embryos in this example, and they freeze 7. They have 2 children, they begin to thaw some more embryos, they build their family of 7 children, and they have 2 frozen embryos left over. Those 2 embryos that were frozen can be discarded, they can be provided to another couple, or they can be donated for research. The other group to think about that is not quite as obvious are individuals that do not have infertility. These are people that are doing in vitro fertilization so that they can do pre-implantation genetic diagnosis of their embryos. These couples usually have either a family member already affected with a single gene disorder, or they know they are carriers for these single gene disorders. So they produce the embryos. In the laboratory, we can take a single cell out of those embryos, test that embryo, and figure out whether it's going to be affected or not affected. The non-affected embryos can be transferred to alleviate having a child with the disease. And in the past, all of these disease-affected embryos have been discarded. And what we have done in the last 5 years is designed a program where we can get donation of these affected embryos that would otherwise be discarded, donated to the University of Michigan under an IRB and fully informed consent, and then work with those affected embryos to make disease-specific embryonic stem cell lines. So why are we using and studying human ES cells as the disease model? First off, we believe that there are biological reasons. Human ES cells are the gold standard. There really is no ethics here. These embryos are being discarded either way. This is a use of an embryo that would have been discarded. In addition, there are reports recently that while there are some genetic and pluripotency similarities between iPS cells and human ES cells, there are still some epigenetic memory within the iPS cells that can influence how they function. With that said, I want to say that I'm a firm believer that using both human ES cells and iPS cells is really what we should focus on, because at the end of the day, we're going to see that some may be beneficial for some types of studies, and another iPS or human ES cell may be beneficial for others. Personally, I would say that there's a reason that I made this decision to focus on human ES cells. One is that, again, as I stated earlier, there really isn't an ethical problem here. There are over 200 single gene disorders that are studied or tested for with pre-implantation genetic diagnosis. Those 200 diseases represent opportunities to make disease-specific models using human ES cells. My personal reason is I was raised in a family of firemen. My grandfather was a fireman, my father was a fireman, my brothers are firemen. Everybody thought I was crazy and that I went to college. But as I went to college and I became a scientist and a PhD, I always wanted to be able to do something that involved patient care. It was not just basic research, but translational. And that's really why 25 years ago, I went into the field of assisted reproductive technologies, working in an IVF laboratory, making embryos to be used to help families with infertility. Well, about 5-6 years ago, I came to a crossroads, and I had done the IVF work for 25 years at the University of Chicago, at the University of Michigan. But I asked myself, is there a way that I can use my skills and knowledge in human embryology, in cell biology, in stem cell biology, to do more good for human health outside of infertility treatment? And when I answered that, I said, yes, we could use our skills for making human embryonic stem cell disease models. So some of our goals is first to derive and distribute human embryonic stem cell lines to the University of Michigan, to researchers outside of the University of Michigan and across the world, to accelerate our understanding of human diseases, develop treatments and cures for these diseases, and really use these human ES cell models to, one, understand how the disease forms, how it progresses, and then be able to use these human ES cell models for screening of pharmaceuticals. And lastly, to engage and educate the public and the research communities, because I believe when you talk to researchers, there's a lot of unknowns with regards to what they think they know and what they really know with regards to human embryonic stem cell disease models. So over the last 5 years, we've worked quite diligently. We now have 13 human ES cell disease models on the NIH registry. We have the largest number in the institutions within the United States. And I'll give some examples of some of these disease models that we've developed. Here you can see the listing of these disease models. So we've acquired, tested, and affected embryos for each of these diseases, converted those into human embryonic stem cell lines, characterized those human embryonic stem cell lines so we know that they function as human ES cell lines and also contain the mutation that gives rise to the disease. Then we submit it to the NIH and place it on the registry. Once it's on the registry, any researcher can purchase those from the University of Michigan, work on those stem cell lines. So there's a couple I'm going to mention here in a moment. The ones that are highlighted in yellow are the only disease-specific embryonic stem cell lines for that disease on the NIH registry. So you can see almost half of those are unique with regards to representing the disease. So a few examples of collaborations. First, for a disease called hypertrophic cardiomyopathy, a mutation in myosin-binding protein C3, we made a human ES cell line. From that, we can direct the differentiation towards cardiomyocytes. We have a very robust method of doing this. We get about 96% purity of cardiomyocytes, and then we can look at the morphology, the function, the organization of those cardiomyocytes when they come from a normal ES cell line, as well as a disease-specific ES cell line. We've also done the same thing for adrenal leukodystrophy. Many of you may know this is the disease that was represented in Lorenzo's oil. The CEO of the ALD Foundation helped us source these embryos. We made the human ES cell line. We're now studying that ES cell line to try to understand this disease better. Another couple examples here are for Fragile X, as well as spinal cerebral ataxia 3. We've made human ES cell lines for both of these diseases. We work on differentiating these into neurons and studying how these diseases develop and progress. Again, the important thing in these studies is that we don't do them alone. We do them together with colleagues that have expertise within the disease. So we take our stem cell expertise, group it together with the disease expertise, and really believe that we're making some great progress. Just giving you an example, we've studied this cardiomyopathy, hypertrophic cardiomyopathy line. We can convert it into cardiomyocytes. The cardiomyocytes are larger when they come from the disease line. They beat with arrhythmia, they beat faster, their sarcomeres are disorganized, and we've also been able to do adenovirus correction, where we correct the gene in the stem cell line with the mutation and demonstrate that the cells are smaller after we do this, and their sarcomeres are more organized, and they beat with a normal frequency. The last thing I'll mention is differentiation of neurons. So again, as discussed earlier, having the stem cell line alone is not that beneficial. Having the stem cell line directing its differentiation to the cells that you're interested in, and then studying and characterizing the disease in those cells is where we will learn a lot. So here you can see this is using the this CSA3 line. We were able to differentiate it to very distinct neural bundles after about 36 days. This is a little different protocol than that was discussed earlier. This is not using embryoid bodies, but it's going directly from human ES cells into neuroepithelium. But this allows us to be able to study these neurons. I'll pass this slide up for time. So I think in conclusion, the important thing to recognize is disease-specific human ES cells really can function as a resource for us to study many different types of single gene disorders. They actually act like a trunk of a tree. One line is for one disease, but we have the opportunity to make numerous human ES cell lines from human embryos that are tested and affected and otherwise would be discarded. So with that, I'd like to reference our webpage that you can look at for more information, and also make a reference to funding. In this area of human ES cell, very different than iPS cell, we cannot, it is not allowed for federal funds to support our research. So in our derivation research, we have to rely on funds from philanthropy, from foundations, and from the local university. Once we put them on the registry, anybody can use those lines and get federal funds to work on them, but we cannot make them with federal funds. And for this reason, I thank Alfred Taubman, a philanthropist, the president of our university, President Coleman, the dean of the medical school, Eva Feldman, who is a neurologist who has been a great push for this, as well as my chair in OBGYN, Tim Johnson, and the EVPMAs of the hospital. Thank you for your attention.
So we have about 15 minutes for questions. Actually, I invite the panelists to join me. And of course, please ask questions. Again, perhaps the first message here is really so surprising to see the progress that medicine has made in just a few years. Because if you recall, 5 years ago, 6 years ago, even more than 10 years ago, these types of modeling was not possible. And now we have the possibility suddenly, not only to generate healthy human tissues for toxicological screenings, but also disease tissues. And when you know that in pharmaceutical research, access to good tissue samples is always extremely hard. So we have that capability right now. So I will welcome questions from the audience, but the first question perhaps to the panelists, what is the limit of this technology? And let's start with Gary, and in this order.
So limits of the technology. I believe you touched on the advantages of the technology. In essence, this removes the need for an animal model. It takes us straight to a human model. I think there's benefits of iPS cells that we don't have with human ES cells, such as the patient specificness of the cells, but we also have advantages with human ES cells when you think about covariates that might be involved and also some of the developmental things. Tim talked about looking at development of the heart. This is one of the things that we've done with our cell line too, and actually been able to identify some of the gene expression issues that occur during cardiomyocyte development in time that we didn't know about with hypertrophic cardiomyopathy. Most people think of that as an adult onset disease, but we've actually found that very early on in cardiomyocyte development that they actually demonstrate some markers of their ability to have arrhythmia and beat fast. So I think those are some of the advantages. I think if we think about human ES cells, limitation is money and people. All of these things take time and all of these things take money. We talked about 200 diseases. One stem cell line per disease is not enough. What we want to do is have 5, 6 stem cell lines per disease so you can do replicates. The other neat thing that we can do that we're starting to do with human ES cells is actually be able to take and do genetically linked stem cell models. So we can make a human embryonic stem cell for a disease, and then we can take fibroblasts from both of the parents of the embryo and from some of the grandparents. And some of the diseases we're just focusing on, the grandparents may have the disease and yet the family does not, and we can begin to look at similarities and differences in those stem cell lines.
Maybe I'll just add a couple limitations with the things that we work on. We're keenly aware of, is it a cell autonomous or a non-cell autonomous defect that we're trying to model? And what I mean by that is if there's an inherent cardiomyocyte defect, we can maybe model it pretty well with our system. But when you get to the three-dimensional structure, the endothelium, the smooth muscle, the vasculature working together, there's where the cell culture by itself in a 2D culture may be a significant limitation when it comes to congenital heart disease. Now, we found similar things that if you look at the gene expression profile of these cells molecularly, you can actually see defects in the cells even in two-dimensional cultures. But, you know, we're not creating a hypoplastic left heart, a three-dimensional model, and I think an opportunity is to better connect with 3D printing, bioengineering, and creating more of a realistic multicellular interface to better model these diseases. Another limitation is just the reproducibility between clones and patients, and we have to be very honest with ourselves with this technology that one clone by itself it could give you an artifact. There could be epigenetic factors, there could be acquired mutations that lead to an artifact when you study one line, and so you really need to be very careful with these studies to have quality control of your cells that you start with and have a selection of multiple clones, multiple patients. Just like we would in any clinical trial, you test drugs on multiple patients, not just on one patient. We all know patients respond differently to drugs, so it's the same thing when you're treating cells of patients. You have to treat multiple patients to have a good statistical study, and I think that is underrepresented right now in the literature because of the cost of doing this work, and one challenge for us is to find ways to make this cheaper and make it more scalable so we can have a broader, more accurate input.
Well, I agree that quality controls are essential because there are variability between the iPS clones, although the technique is very robust. But I think one of the main limitations now is not on the iPS production, but on the differentiation of that cell to the specific cell type. Especially there are some cell types that are not so easy or efficiently differentiated in culture. Only recently, beta cells from pancreas have been robustly differentiated. So cardiomyocytes and neuroepithelium are protocols that have been in place for a long time, but some others from the 200 types of cells that one could get in theory from the iPS, they are still developing them. So that's, I think, one of the main limitations.
Okay, thank you. That's very important to really understand the limit of the technology. I think we all sense the potential of this technology. Again, we can explain epilepsy, we can explain rare disease, we can enable small biotechnology companies to develop drugs for orphan disease because now they have access to a resource that is absolutely critical, which is the right material. We have pharmaceutical companies deploying all these models internally now. And it may help also in terms of their productivity, meaning the number of drugs that can reach the market and provide drugs to patients. I would like to see first if we have questions from our audience. Yes, over there.
Yeah, I have a couple of questions, one for speaker one and a couple of questions for speaker, the one on epilepsy. Sorry, I don't know your names, or can't remember. The speaker one, you mentioned that you could get some effects with beta blockers on the beating of the cardiomyocytes that you produce. I'm wondering how you get that in the absence of a sympathetic innervation. That's my first rather simple question. My two questions for the epilepsy speaker are, did you have glial cells in these? Did you get glial cells in your cultures? Because they're incredibly important in modulating synaptic activity in neuronal cultures. And I was a bit surprised that you suggested you get a reduction in synaptic contacts in a preparation that you're in a sense modeling an epilepsy syndrome, which is hyperexcitability. So I wondered if you could comment on that. Thank you.
Yeah, and so in terms of the cardiac tissue, it's important to point out that these cardiac cultures, I don't care how pure we claim that they are, there's multiple cell types in them. And even if you take a single cell in isolation of a cardiomyocyte as an early stage, there's an autonomous rhythm of contraction of these cells that is pharmacologically modulated at a single cell level of increasing the chronotropic or decreasing it with pharmacologic. Now, it's important to highlight that that doesn't happen in early immature phases of cardiogenesis at first a couple of weeks. It takes 20, 30 days potentially in culture to get the maturity of the cardiomyocytes to express the cell surface receptors to be pharmacologically responsive. So it's not automatic that they are pharmacologically responsive, and it's another degree of maturity that you have to control for to be able to get that.
So at the early stages of differentiation, we get around 5% of astrocytes and less than 1% of oligodendrocytes. We don't get microglial cells because they are coming from another lineage. At the end of the differentiation, we hardly see any of those type of cells. And the second question is very interesting. We are analyzing at the moment which type of cell has these reduced contacts, because we believe that maybe they are GABAergic cells, so the inhibition of a transmission is reduced in this model. One thing that is always surprising us, and that has been also the case for models from Parkinson's disease, is that as differentiation progresses or neurons stay longer in culture, we see different phenotypes, as if the cells were aging. And it's a bit difficult to think in terms, for example, of Parkinson's disease with a 50 or 60-year-old patient. If you leave the cells 30 days in culture, there's no phenotype. If you leave them 75 days, then you see a spontaneous phenotype of, for example, increased sensitivity to oxidative stress. So 45 days in culture somehow allows you to model a disease that takes at least 50 years in a patient. That's really surprising.
Yeah, I completely agree. One interesting observation you should be aware of, maybe you're already aware of, is that the eye cell neurons that Cellular Dynamics actually market these days, they report that something like 70% of their cells are iPSC-derived neurons, 70% of them are GABAergic, and only about 30% of them are glutamatergic, whereas you got the opposite. So that's an interesting sort of difference between two iPSC-derived neuronal cell lines. Thank you.
Well, excellent question. Thank you very much. Do we have another question from the audience? Actually, I have more questions for our panel. So we looked again at the limitations of the technology. Now, I would like to look at the potential of the technology. How much more, how much further can we go in our understanding of the pathobiology of disease, of many indications that have been a big problem? And let's start with cardiovascular indications, and Tim, for you about neurological indications. But what can we dream that is possible now that was not possible yesterday?
So what is possible today with the congenital heart disease focus is using the genotype-phenotype relationships with whole genome sequencing and finding candidate genes. we can actually find specific candidate genes in a family that's disease-causing for this condition of congenital heart disease. And when we model that in culture, we can actually find pharmacological strategies to target that molecular defect. And without genetically correcting that lesion that we can find drugs that compensate for that. And so the future of this goes beyond disease modeling. It goes beyond even understanding the defective process of cardiogenesis. It really goes to individualized pharmacological treatments where we could anticipate what the problem is and correct it with a drug therapy that may be even commercially available, but would be a targeted therapy for this particular patient with this particular disease, and being able to repurpose drugs and treat, and maybe even in the congenital world is going, now I'm putting my speculative hat on here completely, of even potentially treating mothers that have a fetus with the disease so that we can maybe reverse it or modulate that disease phenotype in utero, would be a really powerful game changer technology that is easy to dream about today, but we'll see if the technology can mature to get us there.
So I'll switch away from the cardiac cell and talk about one of the disease models we're working on, which is adrenoleukodystrophy. And again, this is one that we actually have been able to start looking at. This is a disease where you have accumulation of very long-chain fatty acids, and it compromises myelination, so on and so forth. So we're actually looking at, as we go from human ES cells to neural epithelium to more developed neurons, all the way down to oligodendrocytes, what happens with regards to this accumulation of very long-chain fatty acids. And maybe more importantly, with regards to studying this disease, is how these neural cells from embryonic stem cells interact with cells such as cells from the immune system. Talk about the effects of cytokines, because all of these are believed to be secondary effectors of the disease and the severity of the disease in these individuals. So now we have a model in a laboratory. We can study both the neural cell, but also its interaction with cells like lymphocytes or cells or cytokines that might be influencing the disease itself. So again, it gives us some secondary measures or some secondary ways of looking at a disease and answering questions that we've never been able to do before.
Yeah, I agree with those concepts, and I also think that this technology will not only allow the better understanding of the diseases and the discovery of new drugs, but maybe it can also help at the preclinical level to see if define a specific treatment before going into a patient can have any toxic effect on these cells and increase the probability of success when you're going into clinical trials.
Okay, yeah, very good. So again, I would like to stress the fact that pharmaceutical companies, all the pharmaceutical companies, including the big pharmaceutical companies, all these companies have stem cell research tools in their laboratories. That was, again, not an easy switch, because most of these companies said, well, we don't need these models. We have the mice, we have the rats, we have all the large animal models, we have everything we need, we have dogs, we have ferrets, we have everything. But now the power of this technology has imposed itself, and I think it's quite clear. Even though there are technical limitations, and we need to be aware of what those technical limitations are so we can continue to improve on the science, but there is a fantastic potential, and that potential is to enable small pharmaceutical companies to look into rare disease, to enable large pharmaceutical companies to develop better conventional therapeutics, small molecules, and monoclonal antibodies. I think we need also to thank our politicians, wherever they are, in the US, elsewhere, for making this type of research possible. And I think particularly of the state of Michigan, who passed laws to enable this type of research. So it's really important that the entire community, policymakers, investors, researchers, decision makers in companies really understand the potential of this technology, because we are changing medicine in the field of monoclonals and small molecules, but we're changing it in ways that are difficult to still comprehend. Any question, any last question from the audience? I would like to see if anybody has any burning questions. Okay, just one word. What would you like to achieve in the next 5 years? Fernando.
Well, I think still the best is to come. I think we are surprised every 1 or 2 months from new discoveries, so it's a little difficult to envision what's coming next. But I think we would like to get new drugs for epileptic, anti-epileptic drugs, and also new treatments for Parkinson's disease.
Thank you.
Using the genetic information, we'll be able to prioritize candidate pathways and genes, and we'll begin to repurpose drugs that are able to improve the cardiogenic process of individuals with hypoplastic left heart and may improve our diagnostics, but we think that may also allow us to accelerate therapeutic clinical trials with specific molecules dedicated to specific patients.
So I would say with regards to the human ES cell disease specific models, primarily the importance is demonstrating phenocopying of the disease in a Petri dish, or at least an outcome measure that is representative of the disease. Once that's done, rigorous scientific demonstration of that, publication of that, and then go on to doing some of the screenings. But it's really, I think today we're in an early stage of development in all of these types of studies, and it really is just going to take that demonstration that the disease is phenocopied in a Petri dish when you have the mutation or when the iPS cell comes from a patient.
Okay, just if there is no further question from our audience, I think it has been a fantastic session. It's been a great look into the future, looking again, that is considerate of the difficulties, but the potential is there, and continuing to invest into these technologies will really change the way the pharmaceutical industry works. So let's thank our panel one more time. And again, thank you very much for your attention.